tissue samples.
Should we talk about the various different kind of levels, if you like, or aspects to the gut microbiome? Because you've got the luminal microbiome, then you've got the mucosal surface, and then you've probably got another layer, and you've got the mucins and all that kind of stuff. So talk about what different aspects there are and how they might differ.
Yeah, so there's the mucin is interesting because I think in healthy people in the colon, you have these two inner and outer mucin layers. So the inner layer is largely sterile. It's the outer layer where there's some admixture with bugs. And so the interesting question is, to what extent is that different from the luminal microbiota?
Right.
I think there is some difference, but, well, there's a couple of things up to mind. But so one of the things we were interested in, and we got a grant with George McFarlane, sadly now deceased, but a fantastic microbiologist who was working at that time in Dundee, to look at the possibility of biofilms and also whether particle-attached bacteria differed from luminal bacteria. Mm-hmm. And the person we took on to studentship on that project was Alan Walker, who's now much later rejoined.
Who I was with last week.
Later rejoined our group as a member of staff, but much later on. But at that time, his job was to look at that question. And what we have a paper in Environmental Microbiology where we showed that indeed, if you recover solid material, put it through a series of washes, compare that microbiota with what's in the lumen, there are differences. And the key difference, significant difference, was actually in the ruminococcus group, which I've already alluded to. They tend to be particle associated.
Is that a keystone? I don't know why I'm making this.
Yeah. No, I'm very pleased. I'm very pleased you're mentioning that.
Is that a keystone species?
Ruminococcus bromii, we believe, is keystone. And this, well, I might as well talk about it now. The evidence for that, or what made us think of that, was a study that Alan was involved in. Now, Alan, when he left us, joined the Sanger Institute in Cambridge, which was very fortunate from our point of view because it kept us connected. We weren't able to keep up with the sequencing craze that was happening around the world. And because the Sanger Institute of anywhere in Britain had the resource and the ability to do that. So Alan went straight in there and was able to join in with us in a number of projects. What we were able to do at that point was to take advantage of the Rowett Institute's nutrition expertise, because that's what it did, to help analyze a number of ongoing human studies. And so what they were doing uniquely, and this was Alex Johnston, who's still very active in the Institute, was to run highly controlled nutritional studies where the whole of a person's dietary intake was controlled by our kitchens. Okay, and they weren't,
I understand.
They weren't allowed to cheat.
No, I understand.
The things were checked, So
they stayed over, didn't they? And they were.
Well, they didn't necessarily live on site, but they had to report. If they didn't, they had to report to the.
Because I've seen nowadays in the newer Rowett, if I call it that, they can live in the Rowett.
Yes, they could. They could
to make sure they're not sneaking off and having
It depends on the study.
An iron brew and
exactly. It depends on the study whether they. But so this was very unusual. I mean, there's hardly any studies of this type done. So what we were in one of them, a particularly interesting one, what we were able to do, we brought them in for a brief maintenance phase where their diets were all made the same, and then we did a crossover trial in which some of them went on a high resistant starch intake, some on a high wheat bran. In other words, non-starch fiber intake.
Why was it important to look at starch and non-starch? What's the importance of starch for the microbiome?
Well, so there's a medical reason, which is that high starch intake had been linked with certain benefits, and the high fiber intake had been linked with certain other benefits. So the physiologists wanted to know, nutritionists wanted to know what difference that would make to the metabolome and to health indicators.
Got it.
But it allowed us to ask a very interesting question about what differences it make to the microbiome, because at that time there was a general assumption that an individual's microbiome was pretty much fixed. Okay, it will wobble up and down a bit, but and maybe even there were differences between people that had nothing to do with diet. This was the enterotype idea that was around at the time.
And is that enterotype idea dead now? Have we You're going to come to it.
I'll come to that. It's partially dead, but not entirely. But I'll come to that. Okay. But the thing, the dietary trial, what that particular one showed us was that within a few days of switching the diet, you got dramatic increases, not in all species, in certain species, ones that specialized in that substrate. Ruminococcus bromii was the biggest beneficiary from a resistant starch diet switch.
And that changed, just for the listeners, because they're eating up what you're feeding it?
You're providing them with a substrate. That's what we call, what they eat, what the bugs eat.
You're providing them with dinner.
With dinner, which they like. Exactly right. Yeah.